Hello viewers, our next session is on inverse PCR. So the name itself means the PCR product or the priming or the polymerization is in both primer it is taking place in opposite direction. So in a inverse PCR you are using a double stranded DNA and that double stranded DNA is denatured and you are adding a forward primer and reverse primer. But unlike in the normal PCR, these two primers will be uh, priming in within the bordered region. But here it's actually going in the opposite direction. So this is generally this inverse PCR is generally used to make template for sequencing unknown DNA sequence without primer walking. So in the primer walking, suppose if you have an unknown DNA, first you will do a Sanger sequencing. Then based on that sequence, again you will prepare a primer, and again you will do a sequencing. So you need to prepare several primers for sequencing a unknown DNA fragment. But if you are using an inverse PCR, you can have one non-DNA fragment, and using that non-DNA fragment, you can sequence a larger DNA fragment without primer walking. So generally, for that purpose, people will use a transposon. Transposon. So we will be knowing the sequence of that transposon. So this transposon will be introduced into the DNA either by a in vitro reaction or by a in vivo reaction. So these transposons will be introduced into the template DNA. Then after that template DNA can be subjected to restriction digestion. So this restriction digestion you can do either using two different restriction enzyme or you can use a single restriction enzyme. So you should definitely use a restriction enzyme which make a cut within this transposon sequence. Okay. So within this transposon sequence. So the same enzyme can be used for digesting the genomic DNA portion also. So if you do it, you will get a fragment like this for this region. Okay. Similarly, you will get a fragment for this region also. Okay. So you will be getting two different fragments. So one is on the right side of the transposon and another is on the left side of the transposon. You will be getting two fragments. So this fragment you can subject to self-ligation. Okay, you can subject to self-ligation using a ligase enzyme. Either you can clone it into a vector and you can use it, and or you can self-ligate it and directly you can use it for as a template. So generally what we can do is you can self-ligate it and directly use it without ligating with a vector. So, so you will get a ligated product like this, which will be circular, and this circular DNA, it cannot self-replicate because it is not having any origin of replication like a plasmid. So you will be having a fragment like this. So and also you make sure that the template what you are using will have only one this region. Okay, transposon region, only one region. Then after that, you do a PCR. So when you do the PCR, you know the sequence of the transposon sequence. So on the right side of the restriction site, what we have used, you know the sequence. So based on that, what you can do is you can design a primer, okay, two primers, and these two primers should not have any base complementarity or overlapping. So you do a forward, you prepare a forward primer and add to the reaction, and also you prepare a reverse primer and add to the reaction. Add MgCl2, Taq polymerase, dNTPS and MgCl2 and go for the extension step And when you do the extension, so this 3' end will be extending and it will make the product like this, okay, and this also will be extending and will make the product like this. When it is doing it, what you can see is always there will be a nick will be there, because the Taq polymerase cannot ligate it, so there will be a nick will be there. So when you go for the PCR product, so this will be released, this PCR product will be released as a linear structure only. So from a small amount of template like this, you can have several copies of this PCR product. And if you see this PCR product, on either end of this unknown DNA fragment, okay, on either side of the unknown DNA fragment, you have introduced the transposon sequence. Okay, the right side of the transposon sequence, right side of the restriction side of the transposon sequence, or the left side of the restriction side of transposon sequence. So now what you can do is, you know this primer, you have the forward primer and reverse primer. This PCR product you can purify it, or you can ligate with a vector and purify the plasmid, and you can sequence it, or you can directly sequence this product. You can directly sequence this product. You use this forward primer and this reverse primer, so you can easily get the sequence of this unknown region. So that is how inverse PCR is done. So this inverse PCR, now because of next generation sequencing method and all those things, the sequencing of unknown genome or DNA sequence is very easy. So because of that, the inverse PCR very rarely people are doing for transposon mutagenesis analysis and like that. Otherwise, this inverse PCR is also used for introducing site directed mutagenesis. Site-directed mutagenesis. So this site-directed mutagenesis in the overlap extension PCR, per overlap extension PCR, you can introduce only one site modified. But if you are using this inverse PCR, you can have any number of primers with mutated primers. So there, what you are in the overlap extension PCR, what we have seen is you have a mutated forward primer and you have a mutated reverse primer. So for suppose if you are planning for 9 sites, you need 18 oligonucleotides. But if you are using an inverse PCR, what you can see is you need only total 9 oligos, and you can get mutations. And when you get the mutations also, the thing is, you can have a fragment from this one, okay, and you have a template where only one mutation is introduced, either from 1 or from the 2 or from the 3 4 or 5 or 6 or 7 or 8 or 9, like that. So like that, you can have a product with a mutation introduced only at one site, okay. And also you can have a product like you have a mutation 1 introduced, you have mutation 2 introduced, then after that you have a 1 and 2 introduced, or you have 1 and 3 introduced, or you have 1 and 4 introduced, or you have 1 and 5 introduced, or you have 1, 2, 3 introduced, or 1, 2, 4 introduced, or 1, 4, 5 introduced, or 1, 3, 4 introduced, like that, or in combination of 1, 6, 3 or 1, 9, like that. So you can have a PCR product with all the combinations, okay, with all the combinations. So in different combinations you will get it. So you will be having a heterogeneous PCR products where you have one site modified or two site modified or three site modified or four site modified or five site modified or 6, 7, 8 or 9 sites modified. So you will be having all the combinations. So these products you can ligate with a cloning vector, and how you can do a screening, and each plasmid, if you sequence it, you will get all these different types of mutations. So you can study a mutation effect, whether it will affect it because of the mutation at the one region or two region or 3, 4, 5, 6, 7, 8, 9 individually, or one or two combination, or one or three combination, one or four combination. So in all these possible combinations you can study that particular gene, how the mutation is affecting. So that is how the inverse PCR is utilized for different applications. So thank you for listening. So in the next video, we will be discussing about the asymmetric PCR.